Hello everyone, welcome back for more biotechnology. Last time we talked about some of the common applications of using synthesized oligonucleotides. Mainly the thing that we talked about was designing PCR primers. So now that we've got some experience doing that, let's actually spend this video talking about PCR itself. So we talked in a previous video about how complicated in vivo DNA synthesis is, and we also spent some time, thankfully, detailing that in vitro DNA synthesis is a little bit simpler because there's a lot fewer enzymes involved. So what PCR is, or polymerase chain reaction, what that is just a very common and very convenient form of in vitro DNA synthesis that allows us to choose a targeted sequence of DNA that we want to amplify by making millions and millions and millions of copies of that particular sequence. And the way that we do that is by designing the PCR primers, both the forward and reverse primers that we did in the previous video. So PCR is going to require all the different components of in vitro DNA synthesis that we detailed before, and that includes both template DNA, the primers that we designed, DNA polymerase, buffer, and so on and so forth, DNTPs, everything that we talked about before. So what we're going to do is we're going to mix all the components into a very small tube, and there will be a picture of one of these tubes for you to look at later. The reaction volume is pretty small. It's usually somewhere in the neighborhood of 20 to 50 microliters. So once we have all the components in the tube, we are going to place the tube in a device called a thermal cycler. So what the thermal cycler is going to do, the name suggests this, is it is going to rapidly change the temperature so that we can get all the different parts of the DNA synthesis to occur. So our first goal, once we set the tube in the thermal cycler, our first goal is to denature the template DNA. The word denature here means something slightly different than what we said when we talked about denaturing proteins. When DNA is denatured, the two complementary strands break apart from each other because at such a high temperature, like 95 degrees Celsius, the hydrogen bonds that hold the complementary bases together can no longer stay together. So getting the temperature up to 95 degrees Celsius ensures that any complementary base pairing can no longer occur, and we essentially peel the two strands of DNA apart from each other. So what our next goal is, now that we have our two strands peeled apart, is that we want both our forward and our reverse primers to bind. But okay, if 95 degrees Celsius is too hot for the two strands to bind together, then it's certainly going to be too hot for our primers to bind. So what we're going to do now with the thermal cycler is we are going to reduce the temperature rapidly down to 55 degrees Celsius. This is what is called the annealing temperature. So by cooling the temperature down, we allow both our forward and reverse primers to bind to the ends of the two different strands of DNA so that we can set up our double-stranded DNA junctions that the enzyme DNA polymerase will now be able to recognize. And doing so, we should also prevent the two original strands from binding back together, although that will happen to a certain extent. But the important thing is that we will get our primers to bind to a certain fraction of the total strands. So this is why it was so important to consider the melting temperature of our forward and reverse primers when we were designing them. We didn't want them to be too high because that would cause problems later on when we get to the extension step, and then we didn't want them to be too low because that was going to cause problems with getting them to anneal. So because 55 degrees Celsius is well below the melting temperature of both of our primers, assuming we did get it somewhere in the 60 to 65 degree range, they should be able to bind to the single strands of DNA with complementarity. So now that we have our primers bound to the DNA, our next goal is to get to the extension step. Basically, we want to allow DNA polymerase to recognize the duplex DNA, the bonding between the primer and the actual template DNA. So once DNA polymerase binds, it should be able to utilize the free nucleotides in the mixture to synthesize new DNA. However, there is a problem that we need to consider first. So you should hopefully recall that enzymes, which are usually going to be proteins, are very, very sensitive to changes in temperature. For most enzymes, especially when you're talking about human enzymes, when those enzymes are exposed to very high temperatures, they tend to become denatured and they lose all activity, right? So we've talked before about how human enzymes and then a lot of bacterial enzymes like E. coli have temperature optima of around 37 degrees Celsius, which just so happens to be our internal body temperature in most cases. So this causes a problem for us. Everything that we've been doing so far has been happening at either 95 degrees Celsius or 55 degrees Celsius. So those are very, very, very hot temperatures for a human enzyme. Certainly getting up to 95 degrees Celsius is going to destroy any human DNA polymerase that is in the tube. So what that means for us is that using human DNA polymerase enzyme is just not going to be possible here. We're going to destroy the enzyme before it ever has a chance to do its job. So it is for this reason that we are not going to use DNA polymerase from human cells. Instead, we choose to use a DNA polymerase that comes from an organism called Thermus aquaticus. Thermus aquaticus is a type of archaea bacteria that tends to live in very hot environments like hot springs. So for this reason, this type of DNA polymerase is sometimes called Taq polymerase, Taq standing for Thermus aquaticus. The nice thing about this enzyme is that it has a temperature optimum of around 72 degrees Celsius, certainly much higher than the 37 degrees Celsius that you get with human enzymes. So because this enzyme is much more adapted to working at high temperatures, getting the temperature in the thermal cycler up to 95 degrees is not going to be enough to destroy this enzyme. It is very well heat adapted. So for our extension step, we've gone so far from 95 degrees down to 55 degrees for primer annealing, and our next step is going to take place at around 72 degrees Celsius to allow DNA polymerase to do its job and to do its job well. This is what we call the extension step. So Taq polymerase, this is going to vary from enzyme variant to enzyme variant. So Taq polymerase is literally the enzyme that comes straight from the Thermus aquaticus bacteria. But as you can imagine, and from discussions that we've had throughout the semester, Taq polymerase has been genetically engineered to be more robust, to make fewer errors, to have faster operating times. But just Taq polymerase itself synthesizes DNA at a rate of approximately 1,000 base pairs per second. So genetically engineered Taq polymerase may be faster, it may make fewer errors, so it's going to vary from enzyme to enzyme. But let's just say that this Taq polymerase works at a rate of 1,000 base pairs per second. So every second that you allow the thermal cycler to be at 72 degrees, the polymerase is going to be able to synthesize 1,000 base pairs. And I'm just now realizing this should be 1,000 base pairs per minute, not 1,000 base pairs per second. So it's 1,000 base pairs per minute. So the thermal cycler should be programmed to stay at 72 degrees for long enough for the enzyme to finish replicating the template. So when you're programming the thermal cycler, you want to make sure you program it in such a way so that you're considering how long your template is going to be. How long is your targeted sequence that you want to amplify. So the example that we were using in the primer design example that we detailed in the last video, that insulin gene we were trying to amplify contains 333 base pairs. So that's roughly a third of 1,000. So we're going to need a third of a minute in order to amplify all that. So we need to make sure that we allow the thermal cycler to maintain at 72 degrees Celsius for at least 20 seconds. Usually we want to add a few extra seconds just to be careful. But you see the idea here. If the enzyme synthesizes at 1,000 base pairs per minute, not second, minute, then if our template is 333 base pairs, we need at least 20 seconds to enable the enzyme to synthesize all that. If, for example, we have a gene that is 2,000 base pairs, then we're going to need to allow at least 2 minutes in order for the enzyme to synthesize all that as well. So how long the extension step at 72 degrees takes, that's going to depend on what you are trying to amplify. So what we have described so far, going from 95 degrees to denature the DNA, 55 degrees to allow the primers to anneal, and then finally 72 degrees to allow for DNA polymerase to extend, this represents one PCR cycle. So this should theoretically double the amount of template DNA that is in the tube. So that's not necessarily going to be so helpful. So that is why the thermal cycler should be programmed to repeat that process somewhere in the neighborhood of 30 to 35 times. With every time the cycle repeats, that should double the amount of template DNA that is available. So the idea here is that even if you start with a very, very, very astronomically minute amount of template DNA, letting this cycle 30 to 35 times should give you a bunch of amplified DNA. You start with very little and you end up with quite a lot. Doubling the amount of DNA you have every cycle allows for exponential growth in the amount of template DNA that you actually have on hand. And here's what I was promising before. So since everything's happening virtually and you guys didn't actually get to do this semester, you can actually see what a PCR tube looks like. The PCR tube is the one on the left. You are familiar at this point with what a 1.5 milliliter microcentrifuge tube looks like. It's the one on the right. So PCR tubes aren't going to hold a whole lot more than maybe 200 microliters. And I mentioned that most PCR reactions are usually going to be around 50 microliters. So working with PCR and doing PCR definitely is something that requires very careful and very prodigious talent with working with small volumes and micropipettes. So it's definitely something that takes a lot of practice, because working with small volumes is definitely a lot more difficult than working with large volumes. Another thing you'll notice about the PCR tube is that it has very thin walls, and this is for good reason, because since the walls are very thin, the tube is much less insulated, and it allows for rapid heating and cooling, because the thermal cycler is programmed to cycle back and forth between very hot, cooler, and then somewhere in between, so we definitely need the tube to adapt to those changes and to transfer that heat energy into the molecules inside the tube. So if you're putting together a PCR reaction, just to kind of review everything one last time, it might look something like this. If you're coming up with an ingredient list to put into the tube, you might end up with something that looks like 5 microliters of 10x concentrated reaction buffer. So again, that's going to contain all the stuff that the DNA polymerase is going to need, such as magnesium and DTT. You might have 2 microliters of forward primer, 2 microliters of reverse primer, which are usually kept somewhere around 10 micromolar in concentration, 2 microliters of DNTP stock, 1 microliter of the Taq polymerase, which is usually going to be the very last thing that you add, even though it's not the last thing listed here, 1 microliter of template DNA. You really only need somewhere in the neighborhood of 10 to 50 nanograms of DNA to act as your template, and that is not very much at all. You'll recall that nano means 10 to the minus 9th, so that is not very much DNA at all. And then finally, you'll want enough water to bring the reaction volume up to about 50 microliters and enough to dilute the 10x reaction buffer down to 1x. So 5 microliters of 10x reaction buffer in a total of 50 microliters gets you down to 1x, which is exactly what you want. So once you actually finish the PCR reaction, once the thermal cycler is actually done, usually what you're going to want to do is run out your PCR product on an agarose gel. So if you've got 50 microliters of PCR reaction in your tube, you really only have to sacrifice maybe 5 microliters of that, and you should be able to see your PCR product on a gel. So whatever you're going to use that PCR product for, you've still got 45 microliters left in the tube at this point, so sacrificing 5 microliters shouldn't really be that big of a deal, just to make sure that everything went okay. So if we run this on an agarose gel, we're going to do all the good stuff that we talked about before. We'll run the PCR product through the gel. We will stain the gel with something like ethidium bromide, and then we can visualize the gel on a visualization device. So Because you should know exactly how many base pairs your PCR product contains, so if we were amplifying the insulin gene, we should expect our PCR product to be 333 base pairs. And really, because it's such a small piece of DNA, it should run true to size on the gel. If you have something like a Hindi 3 restriction digest of the lambda-phage DNA acting as your ladder. So we should be able to tell very easily that whether the PCR reaction happened or not, because we should see a single band that corresponds to what we expect to see. And if you don't see anything, then that indicates that the PCR reaction did not work very well. So interpreting an agarose gel after running a PCR reaction is usually a very simple task. It either clearly worked or it clearly didn't. There's usually not much interpretation to be done. Alright, so that is going to do it for this video, in which we discussed the theory behind PCR and some of the practical aspects of doing PCR. So next time we will talk about some variations and some applications of PCR technology, starting with quantitative real-time PCR. So thanks as always for your attention.